Hi, this is my great honor to introduce my previous work at King's College London in UK. So my innovation is using the computer simulations to guide the drug design. So the drug design what we are using for the drug is the membrane-active peptides. So this is a kind of drug that can target the specific, specific cell membranes with a unique functions. So if you watch the movie Matrix, you will realize there's a simulated world in the movie, and we are doing something similar in a smaller scale. So we use computer simulations to study how the drug interact with the target, the biomarkers. And in this case, because it's a membrane-active peptide, the biomarker is the cell membranes. So the simulation, what we use here is using the molecular dynamic simulations. Molecular dynamic simulations is a type of simulation that can simulate the atom and molecule movements in the atomic scales. So it's like an atomic microscope. And our approach is using the computer to simulate the drug function and also the structure. And then we can use experiment to validate and then compare with our simulations. And then we can extract the knowledge and to do our drug design in the next cycle. And then we can generate a new protein. And then the cycle will be repeated for several iterations until we got the optimized drug candidates. So why this is important? And the reason is because one single peptide, which is a small protein, can have different kind of events in the biological systems. So as you can see, it can be unstructured, it can be structured, and they can aggregate in a solution. And sometimes they can form a functional structure in the cell membrane. So the peptides can be in the water on the interface of the cell membrane, or it can get into the cell membrane and become like a structure. So there are some examples of what we have done with the computer before. So as you can see, these small proteins can interact with the cell membrane, and they can bind onto the surface and then start to fold as a structure, and they can penetrate across the cell membrane and then start to associate with other small proteins, and then become a functional structure in the membrane. And it can conduct water and ions to cross the membrane, and also other functions. And using the computer, we can mutate the sequence of the protein, and then we can predict what the structure and functions will look like in the cell membrane. So in the bottom here, you can see we compare two different sequences. One sequence has a bigger structure, and one has smaller structure, and they both have different functions. So in biological systems, there are many different kinds of cell membrane types. And for example, our body have different organs, and all the cell membranes are different within the organs. So we can use that as an application to cure cancer, because the cancer cells has different membrane compared to the normal cells. So we can use cell membrane as a biomarker. So what we did is we used the molecular dynamic simulations to guide our drug library design. And then we synthesize the drugs, and then we apply them to the high-throughput screening, so we can evaluate the potential candidates from the screening. And then we use the cell tissue to do the experiments, and then we found the drug candidates. And then we use the nano formulation, which means we can make nanoparticles with the drug, and the nanoparticles can make the drug more bioavailable and also more stable to the human for the treatment. So before doing the human clinical trials, we used mouse as an animal model before we moved that to human. And in the mouse, we used the triple-negative breast cancer in the mouse as a model for our study. The reason why we use triple-negative breast cancer is because triple-negative means there's no biomarkers on the cancer cells for this kind of breast cancer. And in this case, we use the cell membrane as a biomarker. And we found that our nanoparticle can eliminate the tumors from the cells, and the mice they can still survive after several months. So this technology has been patented, and currently we are working with our industrial partner to evaluate their safety before we move it to the clinical trials. So the work is still ongoing. And this is my story, and thank you for listening. Thank you.